Effect of linkage on inheritance. Genes which are present on the same chromosome, if they are tightly linked and if they are not far from each other, they will inherit together. Morgan did another experiment to check whether there is any effect of the linkage on the assortment of the alleles or on the segregation of the alleles. He performed another experiment. During this experiment, he used two types of the flies, considering the two phenotypes. One phenotype was that the fly is a gray color. This is the gray color, while the second fly was in black color. The second thing he considered the wings of the flies. In this case, the wings, they are normal, while in case of second fly, the wings, they were vestigial. So he crossed these two types of the flies. As a result of cross between these two flies, all the flies which were appeared during F1 generation, they were dihybrid, containing the gray body as well as normal wings. If we form their alleles, in case of alleles of this fly, that is female fly, the allele will be this one. In case of male fly, the allele will be this one. As a result of cross between these two flies, F1 progeny was appeared. In F1 progeny, that was wild type as well as that has gray body and the normal wings. So all the fly which were appeared during F1 generation have gray body and normal wings. So he performed the test cross. Test cross is a cross between the F1 generation and one of the parent fly. So cross was between this fly and this fly. So as a result of test cross, after the formation of the gametes, in case of eggs, there were 4 different types of the eggs, and in case of sperms, there were a single type of the sperms. When F2 generation as a result of test cross was obtained, that is a result of test cross, there were some flies which were wild type, normal and gray and normal. There were some flies which were black and vestigial like the parental. This is parental, this is parental, but there were some flies which were gray and vestigial, so new phenotype, and black with normal. They were not similar to the parents. So these two types, they were not similar to the parent flies. If we see what ratio Morgan obtained as a result of test cross, if we perform the comparison, if the genes, two genes for the gray color and the normal wings, if they are located on different chromosomes, as a result of the test cross, the ratio between these flies, that should be 1, 1, 1 and 1 if the genes are located on different chromosomes. While on the other hand, if genes are located on the same chromosome, then the ratio should be 1 to 1 between the flies where we have performed the cross. But the results of Morgan, they were 965 of this type flies, 944 of this type, 206 were of this type, and 185 flies were of this type. So the results of the Morgan was not according to the test cross. In case of test cross, if genes they are located on different chromosome, the ratio should be 1, 1, 1 and 1. While if genes are located on the same chromosome, the ratio should be between the progeny 1 to 1. So he got two parental types progeny while there were two different types. Same is here, the test cross. In test cross, that is between this fly and this fly, the ratio was in the test cross should be 575, all of the four, while what Morgan observed, that was 965, 944, 206 and 184. So according to test cross, the ratio should be 1, 1 and 1. Morgan observed the ratio that was different from the test cross. He reasoned that this difference is due to the crossover and the linkage between the two genes that were for the different phenotypes, for the gray body color and for the wings. Morgan reasons that the genes, they are closely related on the same chromosome. They are not far from each other due to the crossover between the genes. He observed this ratio.